Hi class, this is your instructor, Skylar Huff, and welcome now to Chapter 13. This is Gene Expression. In the last chapter, we discussed how the cell accurately replicates the nucleotide sequence in DNA to pass the genetic information, the genetic material, unaltered, to the next generation. The basic features of that process are the same in all cells studied to date, from the simplest of bacterial cells to the most intricate human cells. In the half-century before that discovery, however, scientists struggled to determine what genes were and how they worked. Early studies suggested that some genes contained information that specified the structure of proteins, but how genes and proteins were connected was unclear. With the discovery of the structure of DNA, it became apparent that these molecules could store the information needed to specify the amino acid sequences for the many different proteins a cell needs to carry out its functions. It would take more than a decade of intense investigation by many scientists before a fundamental understanding was reached of how cells convert DNA information into the amino acid sequences of proteins, a process we refer to as gene expression. It would take many more years of intense work to understand that only a small part of the total DNA in eukaryotic cells actually encodes proteins, and that many other parts of the genome contain genes that encode RNA molecules that regulate gene expression. In this chapter, we will examine the early evidence that indicated that many genes specify the structure of proteins. We then consider how DNA governs the phenotype of the organism at the molecular level through the process of gene expression. For protein-coding genes, this process involves a series of steps in which the sequence of bases in DNA specifies the makeup of a cell's proteins. The proteins affect the phenotype in some way, ranging from readily observable physical traits to the subtle changes detectable only at the biochemical level. The first major step in gene expression is transcription, the synthesis of RNA molecules complementary to the DNA. The second major step is translation. So with translation, this is the process in which the RNA acts as a coded template to direct polypeptide synthesis. So let's begin now with gene expression. So looking at this photograph, this just shows you a TEM, a transmission electron micrograph, with RNA molecules that are synthesized as complementary copies of a DNA template. So keep this in mind as we continue on through the chapter. This is from DNA to proteins. So the idea that genes and proteins are connected originated in the early 20th century, shortly after scientists rediscovered Mendel's principles. In the first edition of his book, Inborn Errors of Metabolism, Gerard, a physician and biochemist, discussed a rare genetic disease called alkaptonuria, which has a simple recessive inheritance pattern. So the condition involves a metabolic pathway that breaks down the amino acid tyrosine, ultimately converting it to carbon dioxide and water. An intermediate to this pathway was, of course, homogentic acid, and it accumulates in the urine of affected people, turning it black when exposed to air. So other symptoms of alkaptonuria include the later development of arthritis in men and stones in the prostate gland. So in Gerard's time, scientists knew about enzymes, but did not recognize that they were proteins. So what he proposed is that this rare genetic disease resulted in the absence of an enzyme in the metabolic pathway that breaks down the amino acid tyrosine. So he found out that, of course, that simple recessive inheritance pattern was found to be a mutation of the gene. Secondly, we got to Sumner. So with Sumner in 1926, he purified a different enzyme, urease, and showed it to be a protein. It was the first clear identification of an enzyme as a protein. So in 1946, Sumner was awarded the Nobel Prize in Chemistry for being able to first crystallize an enzyme. So nextly, we get to the 1940s, and this is, of course, is with Beadle and Tatum. So Beadle and Tatum, they developed a new approach to the problem, in that we have this major advance in understanding the relationship between genes and enzymes. So they knew that there was a series of biosynthetic reactions controlled by specific phenotypes, but it was not yet clear whether the genes themselves were acting as enzymes or whether they're controlled by the workings of enzymes in an indirect way. So what they did here was they chose an experimental organism called a fungus, called Neurospora, and they did that for several reasons. So the wild type, or of course, that individual with that normal phenotype. And what I'm getting at is Neurospora is easy to grow in culture, and it manufactures all of its essential biological molecules when grown on a simple growth medium, containing only sugars, salts, and the vitamin biotin. However, mutant neurosporin strains is one that cannot make a substance such as an amino acid, can still grow if the substance is added to the growth medium. So what happened is that had the mutation in only one gene that affected each gene, only affected one enzyme. So I say all of this to say that by way of Beadle and Tatum, they stated that, of course, one gene class codes for one polypeptide or one protein. Please keep that in mind as we continue on. And nextly, class, we got to another scientist. And the next scientist class is Pauling. So Pauling showed that a mutation of a single gene altered the structure of the protein hemoglobin. And that was huge. So that particular mutant form of hemoglobin is associated with the genetic disease sickle cell anemia. And he determined that, class, by looking at the sickle cell hemoglobin and that of the normal hemoglobin differing, class, only by one amino acid. And just keep in mind that there are two types of polypeptide chains, the alpha and the beta units. So scientists therefore extended the definition of a gene to include that one gene class is responsible for one polypeptide. So just keep all of this in mind as we continue through the chapter. So here, class, gene expression is, of course, that series of steps in which information in the sequence of bases in DNA determine the makeup of the cell's protein. So yet and still, class, first things first is we have DNA. And of course, to get more DNA, you go through the process of replication. So from DNA to RNA, class, happens by way of transcription. And then, of course, from RNA to protein happens by way of translation. So everything, class, needed begins as DNA. So the DNA sequences are what determine the protein by way of that sequence of amino acids, but of course it is not directly. So just keep in mind, and I don't even like the word copied here, I like to say that, yes, when a gene that codes for a protein is expressed, that information in DNA is transcribed to RNA. So keeping in mind that RNA is single-stranded, it has the sugar, ribose, and the base uracil classes there in place of thymine. So RNA is what links DNA and protein. And just keep in mind, class, that A will be paired with U, that is Adenine will be paired with uracil. And then, of course, guanine is still going to be paired with cytosine. And I don't mean paired in the sense class that it's going to be double-stranded. Not at all. I just mean base pairing class, going from DNA to RNA. So with that class, keeping in mind that DNA is transcribed into RNA, and this happens by taking information in one language, that called DNA, and then transcribing it into another language, being RNA. So let's get a bit more into what RNA is. So there are differing types of RNA. First things first, class is called messenger RNA, or mRNA. So it's that single RNA strand that carries the information for making the protein. Next up is tRNA, or transfer RNA. So this is that single strand of RNA that folds back on itself into that specific shape. So to help you with this class, it is, I guess I'll do a little drawing here. Let's draw just a bit. So it is this molecule class called tRNA, and we'll see more of this a bit later. But it is that molecule called tRNA class that is responsible for bonding with those amino acids and then bringing those amino acids class to the ribosome, that site class of translation. And then thirdly class is rRNA, which is ribosomal RNA, and this is that globular or structural part of the ribosome that is catalytic functions needed for protein synthesis. So here, the information class that is transcribed into mRNA is what specifies the amino acid sequence of a polypeptide, or that amino acid sequence class of that protein. So this class, as I get to it, is called the process of translation, using what has been transcribed into mRNA to specify the amino acids of that polypeptide. So we have a sequence of 3 consecutive bases in mRNA, and those 3 consecutive bases in mRNA are referred to as being a codon. So here I'm referring to that nucleic acid language, that amino acid language of proteins. So I'm getting to what is known as that triplet code. That triplet code, such as A, U, A, and I will still put here class still 5' to 3' as far as that direction. So adenine, uracil, adenine. So this is that mRNA sequence of 3 consecutive bases in mRNA that specifies 1 amino acid. And I'll even provide another example, such as UC. So here it is, UC, still class 5' to 3'. And this particular amino acid, well, this particular codon would code for the amino acid phenylalanine. And there is no need to know that class from your brain or by heart. This class will be done by use of the codon chart. So one more time, let me write those once again, and I'll write them at the top right hand corner. It is U, and I said C. This is the 3' in, this class is the 5' in. And what I'll do now is get to how I got there. So RNA, just as DNA was, class, will be, of course, read in the 5' to 3' direction. So you will not necessarily do this on the test, but it's important to know how to use the codon chart, and there's no need, class, to memorize this. So on the 5' in, we have a U. So there is the U on the 5' in. Next up, in the middle class, or that second letter, is also a U, because it is UUC. So we now classify C. We now classify C right there on the 3' in. And then, of course, we now are left with class what is known as UUC. And it's right here. And that class, as I mentioned, is phenylalanine, abbreviated class as PHE. So that's how I got to what I gave you in that example. Next up, class, again, just keep in mind that the ribosome is the site of translation. And, of course, with those 64 possible codons, as I just showed you, class, here in the codon chart. Please don't count these. I just told you that. So with that, I'll get down to, I guess, saying one more thing. This mentions the genetic code, so I must also state, class, that the genetic code is universal. And I say the genetic code is universal because, of course, the organisms that have life, even including bacteria class, we all have those same A's, T's, G's, and C's, and of course the A's, U's, G's, and Z's. And I'll get more to that class in the coming chapters. Just keep in mind that it's not something that is unique class to eukaryotes or prokaryotes. It is life class. So now getting back to tRNA. So tRNA, as I mentioned earlier, class is what connects the amino acids and nucleic acids. So with that being stated, what I'll say nextly with tRNA is that it links with a specific amino acid. So I'm just drawing class a tRNA molecule. And what would typically class be attached? Not typically, what is attached is going to be class some amino acid. So I'll just draw this little circle here. And let's just call this MET. And I said MET class, and that's for methionine. But I'll get to that in just a moment. So it says that each tRNA has an anticodon that, of course, has those hydrogen bonds with the mRNA codon by complementary base pairing. So as I continue with this, what I'm saying about this class is that let's just begin with an example. For instance, in the case of getting here from the 5' to 3' direction, let's start with this codon being AUG. AUG class is the start codon. So since that is the start codon class, just keep in mind that the anticodon, that would be their class on this tRNA, and just keep in mind that the anticodon must match class the AUG. So the anticodon class would be UAC, still from the 5' to 3' direction. So that means that this tRNA class would be right there working to add the first amino acid being methionine. So I'll do more with this class as we continue, but this just gives you class an idea that RNA is translated to form the polypeptide by way of what is known as transfer RNA, tRNA. So this gives you class what tRNA looks like, and this here is where the anticodon would be. And then another depiction class, yet again, where the anticodon would be. This in class would have the amino acid, as I showed you, methionine. And then thirdly class, this is it. So in this case class, we have phenylalanine here. And in that case class, we have the codon UUC. Anticodon classes there, A, G, to match, of course, the codon there found in the mRNA drawn class, there for you in purple. So the ribosome class is the site of translation. It's composed of class both a small and large subunit, and for those who are saying I'd like to see it, let's begin now. So this is that small subunit, this class is that large subunit, and I drew it kind of into my text. I apologize for drawing it into my text. There we go. And now I'll do a bit of writing class on top of such. So this class being the small subunit, this class being the large subunit. And with that class, we have the E site, we have the P site, and then we have the A site. So the A site class is there for the attachment. I repeat, is there for the attachment. So what will attach to their class will be, of course, that tRNA molecule, bringing in class, what is known as that amino acid. And will then class shift to the left here to that P site to attach the amino acid, and then ultimately it will leave class via the E site for exit. Excuse me. And with that I do not have drawn class will be right there, what is known as class, that secondary mRNA molecule, still class from that 5' to 3' direction. And we'll do more with that class here shortly. So it just states that it attaches to the 5' end of the mRNA, and it will travel along its class, allowing the tRNAs to attach sequentially to the codons of mRNA by way of the anticodon to check to ensure that the correct amino acid is being brought in. So those amino acids that are carried by tRNAs are positioned in the correct order class by those peptide bonds to form that polypeptide or protein. So this class provides you that overview of the process. So keep in mind, class, that the non-template strand is here, and the template strand, class, that is being transcribed is right there. So if you look closely, class, translation is occurring, class, in that 5' to 3' direction. For instance, what we're about to get to, class, is the process of taking what is NA and transcribing it, class, into that primary sequence of amino acids. But first things first, this, class, is that mRNA strand. So it's AUG, our historic codon, which codes, class, for methionine here. And then, of course, ACU, UGC, and so on. So this is the process. And then once we have that primary sequence, once we have that primary sequence of mRNA, we'll get to that secondary mRNA. And it is that, class, that will be used to form the protein, as you all see here, by way of translation. And that's why it states there, class, polypeptide. So at the polypeptide, class, where you see that, that's why we have what is known as that primary sequence of amino acids. So that's where we're going, class, with the process. So transcription is nothing more, class, than making the mRNA, and this happens in the nucleolus. So just one more time, the mRNA is that linear sequence of those 4 RNA molecules, being cytosine, guanine, uracil, and adenine. So the code is read, class, 1 codon at a time, or 1 triplet at a time, from a fixed starting point that establishes the reading frame for this message. So yet again, class, the genetic code is universal. So everything, class, on Earth, from, of course, the cnidarians, called the jellyfishes, or even, class, the redwood, of course, that what is called the gymnosperm, to bacteria, class, being prokaryotic, and even to us, class, called Homo sapiens. We all, class, have the same A's, T's, G's, and C's. So that genetic code, as I mentioned earlier, class, is universal. So it states, class, the genetic code is redundant. So yes, it's more than one codon that specifies, class, for most amino acids, as you saw, class, as we got to phenylalanine with, of course, UUC. So just keep in mind that this is called the wobble hypothesis, in that the third nucleotide of a tRNA anticodon may sometimes form hydrogen bonds with one or more than one third nucleotide of a codon. So this wobble results in several acceptable forms of base pairing, class, between mRNA and tRNA. So that's why I mentioned, class, this wobble hypothesis there. So now let's continue on with where we're going, class, with this. All right, finally to transcription. So in eukaryotic transcription, most RNA synthesis requires one of 3 RNA polymerases. So to help you with what I'm getting at, it's either going to be RNA polymerase 1, RNA polymerase 2, or RNA polymerase 3. I'll leave it at that for the purposes of this course, and we'll get to the use of such things. So let us now begin the process, class, of transcription using that template strand. Yet and still, class, is anti-parallel. 5' DNA is here. 5' class of the growing RNA strand is there. Let us continue. So upstream versus downstream. So as I mentioned, upstream and downstream, just keep in mind that downstream is toward the 3' end of the RNA, or that 5' end of that DNA strand. So just keep in mind, class, that we're going to be going in this direction, class, always and forever, 5' to 3' direction with RNA. So from upstream to downstream. So upstream, class, is always going to be that 5' end of the mRNA to the 3' end, class, of that template DNA strand. First things first, class, is initiation. So we'll do these same 3, meaning the same 3 being initiation, the second class being elongation, and then thirdly, class, termination. The same 3, class, will happen again with translation. Let's begin now with transcription, the synthesis of mRNA. So with initiation class, we'll begin at that promoter sequence. So this is that nucleotide sequence in which DNA is, of course, to which the RNA polymerase and associated proteins initially bind. So it's sometimes stated class is going to be, I guess you say, getting everything needed class to build the house. So everything needed class to synthesize that mRNA strand. So what happens is the transcription factors attach to the TATA box to determine the direction of transcription will proceed. And in the case you're saying, why did he say TATA box? That's T-A-T-A. Yes, T-A-T-A. And I'm stating that because TATA is part of that promoter sequence. So we'll then class add more transcription factors to get to the process of elongation. So what I'm getting at now is that RNA polymerase is added to the transcription initiation complex. And then we get to elongation, which is the actual making of that RNA molecule. So it still must be read in that 5' to 3' direction. And you get those RNA polymerases, which of course then separates that double helix and then adds those nucleosides to the growing RNA molecule. So each nucleotide is added to the 3' end of the RNA molecule. And of course, the remaining phosphates become part of that sugar phosphate backbone. And the last nucleotide will be, of course, incorporated to that exposed 3' end. So let's now terminate these. So to terminate it class, that just means to end the synthesis class of mRNA. So with termination class, it's happening because of a stop codon. I did say S-T-O-P, because of a stop codon. So I say stop codon, so that's C-O-D-O-N, and as it happens, this stops transcription class, and this happens at the AAUAA. So I say at the AAUAA sequence. At this sequence class, the process ends, and then it releases from the DNA. So thereafter class, we then get to the part known as, well, I'll show this first. So we have that promoteration class in which things begin. Then we get that direction class of mRNA transcription by way of that template strand. So we're now class making the mRNA in red. DNA class is being uncoiled and unwound by RNA polymerase, which of course is there to add those nucleotides. And now class, we have that primary sequence of mRNA there at the bottom. So nextly, after we've now gotten that mRNA transcript, or that primary mRNA, and of course you're seeing 3 of those here. There's 1, there's 2, there is 3. And I say 3 because these class represent 3 different genes, meaning 1 gene, 1 protein. So yes, all of this class is from the DNA. You see being that double helix here. And what I'm saying in saying all of this is that this class represents a gene here, that represented a gene there, and that represented a gene there, all within that DNA, just in 3 separate sections of that DNA molecule. I'll say more about this in just a moment. Well, I can't wait. It's not class all of your DNA that even codes for a protein. It's only specific sequences, specific areas class of the DNA that code for the protein. So let's get now class to those non-coding mRNA sequences. So just keep in mind that it's not all the DNA that codes. And then at the end of each coding sequence is a stop codon that of course stops the process. So now let's get to post-transcriptional modification class. And this happens in eukaryotes. So I'll do this class in 3 different ways. First things first class, I'll begin with, let's go with front-end modification. So with front-end modification class, what is added is called a 5' cap. So the 5' cap class is added to ensure that enzymes in the cytoplasm of the cytosol do not break down the mRNA. The very same way, a poly A tail will be added. I say again, a poly A tail will be added. So the poly A tail is added for the same reason, class, that 5' cap is added. So, of course, it says capping that 5' end. Well, now for that poly A tail, the very same is the case. It's added to ensure that the enzymes in the cytoplasm, which we are still in the nucleus, class, at this moment, but it ensures, class, that those do not degrade or break down the protein. So nextly, class, I'm getting into what is known as middle modification, the third part. So with middle modification, class, so we've already done the 5' end or front end modification. I've already done, class, that back end modification at that 3' end. Well, now, class, to middle modification. So with middle modification, class, this happens to remove non-coding regions or introns. So I say again, we remove here, class, non-coding regions or introns. So it happens, class, by way of spliceosomes. So I say spliceosomes. So what spliceosomes do, class, is that they remove non-coding regions, class, known as introns. So I've highlighted, class, 2 introns there with what I call red. And then I'll get back to those very same 2 here. So those, class, are non-coding regions, but they do not code for the protein. And they must be removed, class, before this mRNA moves from the nucleus, class, into the cytosol. So in eukaryotes, class, both coding regions and no coding regions, class, are transcribed as well as replicated, meaning those non-coding regions, class, are transcribed as well as replicated. However, class, non-coding regions are not translated in eukaryotes because they are removed. So once you remove, class, those introns, those non-coding regions, what you're left with then, class, is nothing more than those coding regions known as exons. So what you're seeing left over, class, are those 1, 2, 3 exons, the actual coding region of the protein. And yes, we still have the 5' cap there and the poly A tail there. So what will then, class, leave the nucleus? What will then, class, leave the nucleus of the cell? And just keep in mind, class, that in purple is the nucleus here. So I say again, what will leave the nucleus is secondary mRNA via those nuclear pores. Via those nuclear pores, it will leave the nucleus. And the reason it can leave the nucleus, class, is because of the presence of what is known as uracil, because uracil is small enough to fit through those nuclear pores, as opposed to, of course, the thymine, which cannot fit through those nuclear pores. So as it leaves nuclear pores, it being the secondary mRNA, it can then, class, be translated into a protein. So I've said all of this, class, to say one more time, class, that the secondary mRNA is now ready for transport to the ribosomes for translation into the protein. So another thing, class, to keep in mind here is that those exons, and I'll go back up, class, I guess I'll grab this color here, the exons. Of course, they're in this, I guess you could say, maybe the A, B, C arrangement. They could be arranged, class, they could be arranged, class, alternatively as B, C, A, or even class C, B, A. And by rearranging, class, those exons, of course, this is just 3 exons for simplicity and teaching purposes, but they can be rearranged, class, into, I would guess you say, almost an infinite number of proteins. And this is why, class, we have what are known as antibodies, meaning upon exposure, class, to some antigen, we, class, synthesize antibodies, and it's all by rearranging, class, those coding regions. So there we have it, class. So I've already gotten, class, to getting from, I guess you would say, primary mRNA to secondary mRNA. So just keep in mind that as much as 75% of that original class transcript, that primary sequence of amino acids, or I'm sorry, that primary mRNA, as much as 75% of that class, of course, must be removed. So those introns class make up a lot of what won't even be used because it's only exons that code for proteins. There you have it. So next up, class is translation. So this is the processes of making the protein, and this occurs at the ribosome class. So it turns mRNA into that primary sequence of amino acids for the making of a protein. And it does this class with the assistance of tRNA. That is what brings in those free amino acids. So with this class, it converts that triplet code amino acid to those 20 amino acids that you all find. So here, class, those amino acids will be joined by peptide bonds to form proteins. So the amino acids are covalently linked to tRNA, and it's all by the amino acyl tRNA synthesis. So it's the anticodon of tRNA that is complementary class to that mRNA codon. And I've already went over this class just moments ago, so let's continue on. So this class shows the ribosome, both the large and small subunits. So if you look closely, class, this is what I drew earlier. So I said again earlier that we'll have the mRNA there, from that, of course, 5' to 3' direction here. And both the large and small subunit class with that exit site, with the attachment site for the tRNA, and the P site class for, of course, those amino acids for that growing class, primary sequence of amino acids. So the P site class binds to the tRNA, as I mentioned earlier. The A site is for the attachment of that amino acid. And then the E site is for the tRNA's class to exit once, of course, the correct amino acid has been delivered. So this shows you, class, we have that leader sequence. And with that leader sequence, we then have, class, what is called that start codon, which is AUG. It won't change. Then, of course, coming in, class, will be what is known as tRNA, bringing class in what is known as methionine. And then, of course, another amino acid will come along by way of tRNA, and then another amino acid class will come on by way of tRNA, and more, and more, and more, until, of course, we are left with what is known as that primary sequence of amino acids that I've depicted here, class, by way of these shapes. So let's get to the process class of initiation, and this class is in eukaryotes. So in eukaryotes, class, initiation is when we have this small subunit that attaches to the 5' cap, and its signal, class, to that large subunit. So the AUG, or start codon, is what brings in the tRNA using the anticodon with methionine attached, starting protein production. So this is that initiation complex, class. So now we're at the second of 3 class with elongation. And just keep in mind that the large subunit was brought in using GTP, which is similar to ATP, and it just uses initiation factors. So we now have methionine class at the P site, P is in Paul, and we, of course, have the A site open for the attachment class of another tRNA. Hence, we're at elongation, which is making the primary sequence of amino acids. So the same class in bacteria as well as eukaryotes. So as it occurs, class, ribosome translocation goes down the mRNA one code at a time using GTP, and it adds single amino acids using tRNA at that open site. And that open P site is what I'm referring to, using GTP. So GTP class is used to make the peptide bonds at the A and P sites. So the rate of addition is controlled by elongation factors. So we have it here, class. So what comes in, class, is a tRNA with an amino acid. Notice, class, we already have 1, 2, 3 amino acids there. So it will move, class, from, of course, the A site to the P site, and it will then, class, transfer over that amino acid, which you all can now see there. Then, of course, by way of the E site, that tRNA will leave, and of course we have that new peptide bond there. So this is the process, class, of elongation. Next, class, is the third of 3, the same as we saw, class, in transcription. So with termination class, it all occurs because of what is known as a stop codon. So here, class, the stop codon is at the A site. So by way of those release factors, what happens here, class, is at the A site, it causes the hydrolysis reaction, and it releases, class, of the protein. So there you have it. The polypeptide chain breaks. So there we have it, class. The protein has been released, that primary sequence of amino acids. And of course, we also release, class, that large and then that small subunit. And it's all because of the release factor, class, you see in green. Right there, class, into that A site. So let's get now, class, to modification. And just keep in mind, class, that just because we have release, this mRNA that is here, class, can be either A, reused, or B, broken down, but it is, class, going to be recycled. It can very well be recycled. So now, class, to modification, and this is called the mRNA by way of translation. So if the primary sequence, class, enters into a chaperone, if it enters into class a chaperone, keep in mind, class, that if in fact it enters a chaperone, protein will stay inside the cell. I repeat, if in fact it enters into the chaperone class, the protein will stay inside the cell. However, class, if in fact that protein, the primary sequence of amino acids, enters into the rough endoplasmic reticulum, if it enters into that rough endoplasmic reticulum, class, then of course that protein will be exported out of the cell. So just keep in mind, class, that it could very well be that polyribosomes are used, and that just means that the mRNA is bound to clusters of ribosomes to make, class, a number of those proteins. So to look at things here, class, this just gives you how things occur, class, between the two. I'll begin, class, on the right-hand side with what is called the eukaryotic cell. So you can see, class, that it's called compartmentalization, meaning inside the nucleus, class, that DNA is going to be replicated. Yes, I said replicated. But also inside the nucleus, transcription, as well as, of course, replication are occurring. So what happens also, class, is that you have RNA processing. That is where we got to modification, where we left their class with what is known as secondary mRNA, which would then leave by way of those nuclear pores for translation, class, to occur in the cytoplasm, the cytosol of the cell, by way of the ribosomes. That's in the eukaryotic cell. However, class, here in the prokaryotic cells, it happens, class, all in the very same area, meaning DNA is replicated, transcribed, and translated, class, all in the same area, because this type of cell has no, of course, membrane-bound organelles. And they don't, class, have any, I guess you say, removal, class, of exons by way of introns, meaning it's no introns, class. I repeat, there are no introns to remove in bacteria or prokaryotes. As you see it, class, there is no modification, class, of the mRNA here. Keep that tidbit in mind. I'll now continue. To mutations, class, and look for these on your exam. Meaning on the test, you all will explain the process. It's the process of what? Transcription, and the process of what? Translation. And I'll also ask questions, class, about mutations. Let's now begin. So mutation, classes, I would say nothing more, class, than nucleotide sequence changes in either A, DNA, or B, mRNA. So if you're wondering what causes mutations, class, well, CO2, or those things called exhaust out of the tailpipe. So please watch out for those. I would say never, class, sit in a vehicle in a closed garage. That could be fatal. Not to mention cause mutations. But of course, chemicals, class, cause mutations. Meaning, I guess you say these are what are known as mutagens. Mutagens, of course, cause mutations. And I've just given you, class, a couple of examples. More examples, class, are things such as viruses. Yes, viruses. Alcohol in excess. Smoke, and I refer to cigarette smoke particularly. And even, class, ultraviolet rays. Those that are, class, natural, or those, class, that are not natural, i.e. coming from a tanning bed. So it states, class, the frequency of damage to DNA is low. It's because organisms have enzyme systems to repair DNA damage. But of course, those that are uncorrected, class, mutations are either silent or harmful. A few are useful. And just keep in mind, class, that mutations provide that variation among individuals that evolutionary forces act on. And we'll get, class, more so to evolution in the coming chapters. So it involves, class, change in one pair of nucleotides, and they can be, class, either A, silent, or B, class, nonsense. And even a missense. So what I'm stating, class, is let's go through these, class, to make sure you understand what I'm saying. So a mutation, class, that is a missense, class, of course, changes the amino acid. It says it right there. A missense mutation changes the wild type. I said wild. It changes the wild type. The wild type, class, is what's normal and what things ought to be, what that amino acid should be. So if it's a missense mutation, it changes the amino acid class from what it should be to, of course, a mutant form, or a form that is not what it ought to be. A silent mutation, class, is one that causes, I guess you would say, a silent mutation causes no change, in that amino acid sequence. And then thirdly, class, a nonsense mutation, class. A nonsense mutation would, class, change what was once, class, an amino acid to what is known as a stop codon. So now, class, these are just regular old base pair substitution mutations. I mean, this is that change, class, in a single base pair. This letter, class, this base changes to that letter, or that base. So let's now continue. Next up, class, frameshift mutations. So with frameshift mutations, this class changes an entire codon sequence. So I say this to say that it alters the entire codon sequence, period. So, of course, it may very well, class, be that called an insertion or a deletion, but it takes out that entire base. And by taking out the entire nucleotide, class, it shifts down the entire frame. So here in A, class, we have what is known as the wild type. This, class, is what should be normal. So if you wanted to just copy this down, that would help, because I'll go through this, class, and show you. I'll go through 5 examples here. So the DNA, class, which is normal. I'll be at this third codon here, the third codon sequence at least, as far as being an RNA. So what we have, class, is AGG. Well, AGG, class, has now been turned into AGA. And because it's AGA, class, in this case, it's a silent point mutation, because what is arginine, class, is still arginine. Up next, class, at C, we have, instead of being ACA, is actually, class, now in the mRNA as ACA. In that, we still, class, don't have that very same amino acid being arginine. In fact, class, we have that amino acid there, the THR. That is a missense. And now, class, here, instead of having, well, this ATC, we have the TAC that is changed from the wild type, giving us, class, what is coded for a stop codon here. Still with arginine, if you're wondering, still with the ASN, but TRP, class, which is here, and the ASP, which is there, are now, class, both gone and been substituted, class, for a stop codon in a nonsense. Up nextly, we have, class, the complete removal, class, of T and G, and of course coding for a stop codon. So that's an extreme nonsense. And then we have, class, what has been taken out, class, meaning a deletion of this T here, because it was once, class, or at least what we should have there, class, is not there anymore. And just by missing, class, this one, I repeat, by missing, class, this one letter here, we now have, class, an extreme Missent. So it shifted the entire frame down by one codon. So please keep in mind that I'll ask about these mutations. This class has been Chapter 13. And this has all been about the discovery class of the gene to protein relationship. Please prepare well for your test class. And this has been your instructor, Skylar Huff.